Protein. Biochem. Working with proteins. Biochemists' understanding of protein structure and function has been derived from the study of many individual proteins. To study a protein in detail, the researcher must be able to separate it from other proteins in pure form, and must have the techniques to determine its properties. The necessary methods come from protein chemistry, a discipline as old as biochemistry itself and one that retains a central position in biochemical research. Proteins can be separated and purified. A pure preparation is essential before a protein's properties and activities can be determined. Given that cells contain thousands of different kinds of proteins, how can one protein be purified? Classical methods for separating proteins take advantage of properties that vary from one protein to the next, including size, charge, and binding properties. These have been supplemented in recent decades by methods involving DNA cloning and genome sequencing that can simplify the process of protein purification. The newer methods, presented in chapters 8 and 9, often artificially modify the protein being purified, adding a few or many amino acid residues to one or both ends. Convenience thus comes at the price of potentially altering the activity of the purified protein. The purification of proteins in their native state, the form in which they function in the cell, usually relies on methods described here. The source of a protein is generally tissue or microbial cells. The first step in any protein purification procedure is to break open these cells, releasing their proteins into a solution called a crude extract. If necessary, differential centrifugation can be used to prepare subcellular fractions, or to isolate specific organelles, see Fig. 1-9. Once the extract or organelle preparation is ready, various methods are available for purifying one or more of the proteins it contains. Commonly, the extract is subjected to treatments that separate the proteins into different fractions based on a property such as size or charge, a process referred to as fractionation. Early fractionation steps in a purification utilize differences in protein solubility, which is a complex function of pH, temperature, salt concentration, and other factors. The solubility of proteins is lowered in the presence of some salts, an effect called salting out. The addition of certain salts in the right amount can selectively precipitate some proteins, while others remain in solution. Ammonium sulfate, NH4, 2SO4, is particularly effective, and is often used to salt out proteins. The proteins thus precipitated are removed from those remaining in solution by low-speed centrifugation. A solution containing the protein of interest usually must be further altered before subsequent purification steps are possible. For example, dialysis is a procedure that separates proteins from small solutes by taking advantage of the protein's larger size. The partially purified extract is placed in a bag or tube made of a semi-permeable membrane. When this is suspended in a much larger volume of buffered solution of appropriate ionic strength, the membrane allows the exchange of salt and buffer, but not proteins. Thus dialysis retains large proteins within the membranous bag or tube, while allowing the concentration of other solutes in the protein preparation to change until they come into equilibrium with the solution outside the membrane. Dialysis might be used, for example, to remove ammonium sulfate from the protein preparation. The most powerful methods for fractionating proteins make use of column chromatography, which takes advantage of differences in protein charge, size, binding affinity, and other properties, Fig. 3-16. A porous solid material with appropriate chemical properties, the stationary phase, is held in a column, and a buffered solution, the mobile phase, migrates through it. The protein, dissolved in the same buffered solution that was used to establish the mobile phase, is layered on the top of the column. The protein then percolates through the solid matrix as an ever-expanding band within the larger mobile phase. Individual proteins migrate faster or more slowly through the column, depending on their properties. Ion exchange chromatography exploits differences in the sign and magnitude of the net electric charge of proteins at a given pH, Fig. 3-17a. The column matrix is a synthetic polymer, resin, containing bound charged groups. Those with bound anionic groups are called cation exchangers, and those with bound cationic groups are called anion exchangers. The affinity of each protein for the charged groups on the column is affected by the pH, which determines the ionization state of the molecule and the concentration of competing free salt ions in the surrounding solution. Separation can be optimized by gradually changing the pH and/or salt concentration of the mobile phase so as to create a pH or salt gradient. In cation exchange chromatography, the solid matrix has negatively charged groups. In the mobile phase, proteins with a net positive charge migrate through the matrix more slowly than those with a net negative charge, because the migration of the former is retarded more by interaction with the stationary phase. Figure 3-16 layered on. In ion exchange columns, the expansion of the protein band in the mobile phase, the protein solution, is caused both by separation of proteins with different properties and by diffusional spreading. As the length of the column increases, the resolution of two types of protein with different net charges generally improves. However, the rate at which the protein solution can flow through the column usually decreases with column length. And as the length of time spent on the column increases, the resolution can decline as a result of diffusional spreading within each protein band. As the protein-containing solution exits a column, successive portions, fractions, of this effluent are collected in test tubes. Each fraction can be tested for the presence of the protein of interest as well as other properties, such as ionic strength or total protein concentration. All fractions positive for the protein of interest can be combined as the product of this chromatographic step of the protein. Purification. Figure 3. Purification and magnet exclusion. According to worked example, peptide, a bio solution. Figure 3-17 shows two other variations of column chromatography in addition to ion exchange. Size exclusion chromatography, also called gel filtration, Fig. 3-17b, separates proteins according to size. In this method, large proteins emerge from the column sooner than small ones, a somewhat counterintuitive result. The solid phase consists of cross-linked polymer beads with engineered pores or cavities of a particular size. Large proteins cannot enter the cavities and so take a shorter, and more rapid, path through the column, around the beads. Small proteins enter the cavities and are slowed by their more labyrinthine path through the column. Size exclusion chromatography can also be used to approximate the size of a protein being purified, using methods similar to those described in Figure 3-19. Affinity chromatography is based on binding affinity, Fig. 3-17c. The beads in the column have a covalently attached chemical group called a ligand, a group or molecule that binds to a macromolecule such as a protein. When a protein mixture is added to the column, any protein with affinity for this ligand binds to the beads, and its migration through the matrix is retarded. For example, if the biological function of a protein involves binding to ATP, then attaching a molecule that resembles ATP to the beads in the column creates an affinity matrix that can help purify the protein. As the protein solution moves through the column, ATP-binding proteins, including the protein of interest, bind to the matrix. After proteins that do not bind are washed through the column, the bound protein is eluted by a solution containing either a high concentration of salt or free ligand, in this case, ATP, or an analog of ATP. Salt weakens the binding of the protein to the immobilized ligand, interfering with ionic interactions. Free ligand competes with the ligand attached to the beads, releasing the protein from the matrix. The protein product that elutes from the column is often bound to the ligand used to elute it. Chromatographic methods are typically enhanced by the use of Hplc, or high-performance liquid chromatography. Hplc makes use of high pressure pumps that speed the movement of the protein molecules down the column, as well as higher quality chromatographic materials that can withstand the crushing force of the pressurized flow. By reducing the transit time on the column, Hplc can limit diffusional spreading of protein bands and thus greatly improve resolution. The approach to purification of a protein that has not previously been isolated is guided both by established precedents and by common sense. In most cases, several different methods must be used sequentially to purify a protein completely, each method separating proteins on the basis of different properties. For example, if one step separates ATP-binding proteins from those that do not bind ATP, then the next step must separate the various ATP-binding proteins on the basis of size or charge to isolate the particular protein that is wanted. The choice of methods is somewhat empirical, and many strategies may be tried before the most effective one is found. Trial and error can often be minimized by basing the new procedure on purification techniques developed for similar proteins. Published purification protocols are available for many thousands of proteins. Common sense dictates that inexpensive procedures such as salting out be used first, when the total volume and the number of contaminants are greatest. Chromatographic methods are often impractical at early stages, because the amount of chromatographic medium needed increases with sample size. As each purification step is completed, the sample size generally becomes smaller. Table 3-5, making it feasible to use more sophisticated proteins can be separated and characterized by proteins can be separated and characterized by Table 3-5, making it feasible to use more sophisticated and expensive chromatographic procedures at later stages. Proteins can be separated and characterized by electrophoresis. Another important technique for the separation of proteins is based on the migration of charged proteins in an electric field, a process called electrophoresis. These procedures are not generally used to purify proteins, because simpler alternatives are usually available and electrophoretic methods often adversely affect the structure, and thus the function of proteins. However, as an analytical method, electrophoresis is extremely important. Its advantage is that proteins can be visualized as well as separated, permitting a researcher to estimate quickly the number of different proteins in a mixture or the degree of purity of a particular protein preparation. Also, electrophoresis can be used to determine crucial properties of a protein such as its isoelectric point and approximate molecular weight. Table 3. Enzyme. Procedure. Fraction 1, 2, 3, 4, 81, 5. Affinity Chromatography. Note: Figure 3. Electrophoresis of proteins is generally carried out in gels made up of the cross-linked polymer polyacrylamide, Fig. 3-18. The polyacrylamide gel acts as a molecular sieve, slowing the migration of proteins approximately in proportion to their charge-to-mass ratio. Migration may also be affected by protein shape. In electrophoresis, the force moving the macromolecule is the electrical potential, E. The electrophoretic mobility, μ, of a molecule is the ratio of its velocity, v, to the electrical potential. Electrophoretic mobility is also equal to the net charge, z, of the molecule divided by the frictional coefficient, F, which reflects in part a protein's shape. Thus, the migration of a protein in a gel during electrophoresis is therefore a function of its size and its shape. An electrophoretic method commonly employed for estimation of purity and molecular weight makes use of the detergent sodium dodecyl sulfate, SDS, dodecyl denoting a 12-carbon chain. A protein will bind about 1.4 times its weight of SDS, nearly 1 molecule of SDS for each amino acid residue. The bound SDS contributes a large net negative charge, rendering the intrinsic charge of the protein insignificant and conferring on each protein a similar charge-to-mass ratio. In addition, SDS binding partially unfolds proteins, such that most SDS-bound proteins assume a similar rod-like shape. Electrophoresis in the presence of SDS therefore separates proteins almost exclusively on the basis of mass, molecular weight, with smaller polypeptides migrating more rapidly. After electrophoresis, the proteins are visualized by adding a dye such as Kumasi blue, which binds to proteins, but not to the gel itself, Fig. 3-18b. Thus, a researcher can monitor the progress of a protein purification procedure as the number of protein bands visible on the gel decreases after each new fractionation step. When compared with the positions to which proteins of known molecular weight migrate in the gel, the position of an unidentified protein can provide a good approximation of its molecular weight, Fig. 3-19. If the protein has two or more different subunits, the subunits are generally separated by the SDS treatment and a separate band appears for each. Isoelectric focusing is a procedure used to determine the isoelectric point, pi, of a protein, Fig. 3-20. A pH gradient is established by allowing a mixture of low molecular weight organic acids and bases, ampholytes, P81, to distribute themselves in an electric field generated across the gel. When a protein mixture is applied, each protein migrates until it reaches the pH that matches its pi. Proteins with different isoelectric points are thus distributed differently throughout the gel. Combining isoelectric focusing and SDS electrophoresis sequentially in a process called two-dimensional electrophoresis permits the resolution of complex mixtures of proteins, Fig. 3-21. This is a more sensitive analytical method than either electrophoretic method alone. Two-dimensional electrophoresis separates proteins of identical molecular weight that differ in pi, or proteins with similar pi values, but different molecular weights. Figure 3. Subjective. Figure 3. Unseparated proteins can be quantified. To purify a protein, it is essential to have a way of detecting and quantifying that protein in the presence of many other proteins at each stage of the procedure. Often, purification must proceed in the absence of any information about the size and physical properties of the protein, or about the fraction of the total protein mass it represents in the extract. For proteins that are enzymes, the amount in a given solution or tissue extract can be measured or assayed in terms of the catalytic effect the enzyme produces, that is, the increase in the rate at which its substrate is converted to reaction products when the enzyme is present. For this purpose the researcher must know 1. The overall equation of the reaction catalyzed, 2. An analytical procedure for determining the disappearance of the substrate or the appearance of a reaction product, 3. Whether the enzyme requires cofactors such as metal ions or coenzymes, 4. The dependence of the enzyme activity on substrate concentration, 5. The optimum pH, and 6. A temperature zone in which the enzyme is stable and has high activity. Enzymes are usually assayed at their optimum pH, and at some convenient temperature within the range 25 to 38 degrees Celsius. Also, very high substrate concentrations are generally used, so that the initial reaction rate, measured experimentally, is proportional to enzyme concentration, Chapter 6. By international agreement, 1.0 unit of enzyme activity for most enzymes is defined as the amount of enzyme causing transformation of 1.0 mole of substrate to product per minute at 25 degrees Celsius under optimal conditions of measurement. For some enzymes, this definition is inconvenient, and a unit may be defined differently. The term activity refers to the total units of enzyme in a solution. The specific activity is the number of enzyme units per milligram of total protein, Fig. 3-22. The specific activity is a measure of enzyme purity. It increases during purification of an enzyme and becomes maximal and constant when the enzyme is pure, Table 3-5. Figure 3-20. After each Figure 3 After each purification step, the activity of the preparation, in units of enzyme activity, is assayed. The total amount of protein is determined independently, and the ratio of the two gives the specific activity. Activity and total protein generally decrease with each step. Activity decreases because there is always some loss due to inactivation or non-ideal interactions with chromatographic materials or other molecules in the solution. Total protein decreases because the objective is to remove as much unwanted or nonspecific protein as possible. In a successful step, the loss of nonspecific protein is much greater than the loss of activity. Therefore, specific activity increases even as total activity falls. The data are assembled in a purification table similar to Table 3-5. A protein is generally considered pure when further purification steps fail to increase specific activity, and when only a single protein species can be detected, for example by electrophoresis. Figure For proteins that are not enzymes, other quantification methods are required. Transport proteins can be assayed by their binding to the molecule they transport, and hormones and toxins by the biological effect they produce. For example, growth hormones will stimulate the growth of certain cultured cells. Some structural proteins represent such a large fraction of a tissue mass that they can be readily extracted and purified without a functional assay. The approaches are as varied as the proteins themselves. Summary 3.3 Working with proteins. Proteins are separated and purified on the basis of differences in their properties. Proteins can be selectively precipitated by changes in pH or temperature, and particularly by the addition of certain salts. A wide range of chromatographic procedures makes use of differences in size, binding, affinities, charge, and other properties. These include ion exchange, size exclusion, affinity, and high-performance liquid chromatography. Electrophoresis separates proteins on the basis of mass or charge. SDS gel electrophoresis and isoelectric focusing can be used separately or in combination for higher resolution. All purification procedures require a method for quantifying or assaying the protein of interest in the presence of other proteins. Purification can be monitored by assaying specific activity. 3. For the structure of proteins, primary structure.